I don't even know what first got me interested in biology. Like, as far back as I remember, I've had like a weird obsession with becoming a biologist. I'm Cam Brown. I'm finishing up my master's degree at the University of Guelph, and I study environmental DNA and how it impacts aquatic invasive species.
It's always very exciting and rewarding when you find people with a genuine interest and passion for the research we do. So when Cam came to the lab voicing an interest in learning more about how to use eDNA, we were happy to have him join. My name is Bob Hanner. I'm a professor in the Department of Integrative Biology here at the University of Guelph.
Initially, when I went to try and get an undergrad thesis for my fourth year, I was initially rejected. So I was sad for a little bit because I didn't have a project. And then 2 weeks later, I get an email being like, hey, there's this thing called environmental DNA we want to try out. It involves genetics and molecular biology, ecology, and also remote sensing, geographic information system. I do like working on invasive species because I think there's a very practical reason to approach invasive species. What I was looking for was less about a particular kind of species and more about finding actionable problems. And invasive species are one of those things where it's like, okay, we need to figure out how to mitigate the damage.
People need to understand the importance of sea lamprey control because there are profound effects on the ecology of the Great Lakes. And as part of our role as responsible stewards of the environment, we need to develop ways to mitigate their impacts on native species.
I think there's a couple different reasons why people would want to use environmental DNA. Most traditional methods, you're much more impacted by the environment and how you collect them. So I think eDNA is very beneficial for a lot of these things. The traditional way of monitoring sea lamprey or larval sea lamprey is using electrofishing. eDNA removes a lot of the impact of the environment when you're collecting because we can stand on the shore and we can filter water from the shore. That means we can do eDNA sampling year-round, and it also is putting crews and creatures in the river at less of a risk because we're not shocking at random trying to collect. Basically how it works is we collect a sample in the field and we take it through this entire process where at the very end we have data that tells us about species occurrence, and we can use these to make sort of on-the-ground conservation calls. So what I really like about what I do is sort of finding the little details in that system to improve it.
My favorite part of science is interacting with young people as we develop knowledge of the environment around us and try to find ways of protecting it. The modern molecular tools that we now have at our disposal are really revolutionizing the way that we can survey biodiversity.